Yeah, if you want to crack the CSIR net as well as if you want to crack the set exam, okay, compulsory remember every one of you my words, even for the JLDL, plant physiology is one of the most important unit that every student has to read a compulsory, okay. So it is very important. Everyone, please mute your mics. Do not make any kind of a noise. Telling you, right? How many times I should tell to all of you like a kids? Okay, I'm telling you, right? I'm always requesting you maintain professionalism when we are in a class. Okay, please do not cause any kind of interruption to the others as well. Okay, firstly, don't cause interruption to me when I am teaching something. It is always disturbs my flow of teaching. Requesting everyone always turn off your mic as well as the video. Okay, all right. Okay. So as I am saying to all of you, plant physiology is one of the most important unit that we have to read without fail. What is the reason? The reason is that this is also like a developmental biology unit where there is a no number of questions that comes bias based. Okay, there is a no bias here. Most of the time you can expect minimum of 20 to 24 marks very surely from the chapter of plant physiology. That's the reason we should not neglect, even though you're from a zoology background, even though you're from a biotechnology, especially microbiology people, they will feel plant physiology is one of the difficult unit. So it doesn't matter what is your specialization, friends, you have to read this particular unit compulsory without skipping it. Now, what are the various type of subunits that we have to cover here? Photosynthesis, in that only respiration, photorespiration will come, nitrogen metabolism, plant hormones, transport in plants, sensory photobiology, secondary metabolites, and stress physiology. Let me tell you one common point. I have seen many students, even though they read this chapter for 1 year, when it comes to the examination, I observed that 70% of the students, even though they are having a well concept in their mind, they are unable to answer the question very correctly. So please mute your mics. The reason may be multiple reasons. Okay. The reason may be, is it because of the difficulty level of the question, or the reason may be, the question will be purely analytical. Don't get confused. So what are the various most important topic you have to focus on? Without this, you should not, you cannot answer even a single question also. One of the major topic will be the photosynthesis. You should not skip under any circumstances. Number two, nitrogen metabolism. Number three, plant hormones, and super important among all, that is transport in plants and secondary metabolites. These five topics are super important. And but I also encourage you, right now, since it is a revision course, I will cover the five topics very briefly and elaboratively. But I want you people majorly focus on stress physiology and sensory photobiology also. In the previous classes we have discussed it. you know I want you to cover those topics as well, but these five topics are highest preference. You have to read those five topics compulsory for the exam. All right, I'll go little speed, try to cope up with this. Okay, what kind of a questions we will get? If I observe last 10 question papers, an example, 7 to 8 times out of 10 question papers, the question comes from 7 to 8 term statement based only. One time they have asked the question experimental, and recently they have asked one question from the graphical part. So that's the reason you have to read this entire topic from basic to the advanced level. So what is actually the process of the photosynthesis? You all people know very well. Photo means what generally? Okay, am I audible to everyone? Quickly confirm, am I audible to everyone? Now clear? All right, all right, all right, all right. Okay. I hope it's clear for everyone. My voice is breaking. I hope it's clear for everyone now. Voice is breaking. Okay. Okay, is it fine now? Okay. Okay. Is it clear now? Now is it clear for everyone? Confirm me quickly, please. All right, all right, all right, all right, all right. All right, all right. Please continue. Please continue. So, what is the process of the photosynthesis? We all people know very well. Photo means light. Synthesis. What is the meaning of a synthesis? We have to make something. We have to prepare something. Now, if I ask you a very simple question, what actually plant will prepare? Plant will prepare its food, because if the plant want to survive, if the plant wants to grow, if the plant wants to develop, plant has to compulsory synthesize its food in the form of a sugar. Now the question is here, how the plant is going to synthesize its food. If the plant want to synthesize its food, it's compulsory require the sunlight, it require the water. So it will take the energy from the sun, it will take the water from the soil, and it will also take carbon dioxide from the atmosphere. Now every student remember, what is the ultimate goal of the photosynthesis? The ultimate goal of the photosynthesis is a reduction of the carbon into sugar. This is also asked a question. Photosynthesis is what kind of a reaction? Is it a reduction or oxidation? Remember, photosynthesis is always the reduction, because we are reducing the carbon into the sugar. Don't get confused. So now every one of you try to understand the point. Whenever plant is preparing its own food, remember everyone, whenever the plant is preparing its own food, as a part of their food development, it is releasing oxygen as a byproduct. It is releasing the oxygen as an end product. So remember my words, no plant on this earth releasing the oxygen purposely. So every plant starts releasing the oxygen as a byproduct or as a end product. Now, we read this equation, very long back. You just remember 6 molecule of carbon dioxide combined with 6 molecules of water and giving rise to 1 molecule of sugar by the release of 6 molecules of oxygen. Just remember 6616. But what is the point? Even though carbon dioxide is present, but what is the major here? Even though light is present, what else is the most important one? Chlorophyll is the most important one. Because friends, who is going to absorb the light energy? Who is going to convert the light energy into the chemical energy? Undoubtedly, the chlorophyll molecule only. That's the reason what we can say that the chlorophyll is extremely important for the process of the photosynthesis. No point of getting confused over there. Now, a very common point I want you to remember, where does the process of photosynthesis occurs? Where does the process of the photosynthesis occur? This is the most important one here. In our competitive exam, always mark my words, most of the questions will come from the locations. So whatever the lesson that you are reading, whenever you come across of any location, it is very important to remember for the exam. Where does the photosynthesis is actually taking place? Remember friends, photosynthesis is actually taking place in an organelle called as the chloroplast. And you all people know very well, if you look into the structure of a chloroplast, how does it look like? Every one of you look into this. Chloroplast is a double membrane organelle. One is called as outer membrane and another one is called as the inner membrane. And you can see the picture very clearly friends. There is a space is located between the outer membrane and inner membrane, which we generally call it as the intermembrane space. No point of getting confused. Very important, if you observe the structure of the chloroplast inside, every one of you focus. Inside the chloroplast, there is a flattened vesicle sac-like structures are present. What do we can call it? There is a flattened vesicle sac-like structures are present inside the chloroplast, which we generally call it as a thylakoid. Every one of you look into the picture, friends. How does the thylakoid is looking like? This is how the thylakoid is actually looking like. You can see the picture very clearly. Correct, na? Now, the most important point here, the thylakoids which are present in the chloroplast, they are not a single. What you people can observe here, the group of thylakoids are arranged in a group. So friends, that group of thylakoids are only what we generally call it as, the granum. And what you people can observe here, you can observe that one group of thylakoids can be connected to the another group of the thylakoids. What? One group of the thylakoids can be connected to the another group of the thylakoid with the help of a thin thread-like structure. So this is a thin thread-like structure. That is what exactly we can call it as the lamella. And what does the what else chloroplast contain? Of course, chloroplasts contain fluid-filled structure. What do we call this particular fluid-filled structure? This fluid-filled structure generally called as the stroma. No point of getting confused over there. And you all people commonly know that the chloroplast is involved in the synthesis of a protein. It is involved in what? Synthesis of a protein. So what we can say that, so if they want to synthesis, they require the ribosome, which is a type of a 70S. And what else it contain? I believe every student remember my words. We generally use it to say that chloroplast and mitochondria are the self-replicating organelle and semi-autonomous organelle, which means that they can synthesize their proteins on their own. Correct, na? So why they can synthesize their proteins on their own? Because they are having a specific DNA. Now, the question which is asked in the AP set exam, and the basic level question asked in day before yesterday, NEET exam also. The type of a DNA that is present in the chloroplast. Every student remember the type of a DNA present in the chloroplast is the double-stranded DNA, which is a circular DNA. There is no point of getting confused here. The DNA is a completely circular DNA. Very important statement to be remembered for the exam. All right. Now, I'll quickly summarize whatever I have explained to all of you friends. If you observe the structure of the chloroplast is a double membrane organelle which contain the inner membrane, which contain the outer membrane. The space between the inner and outer is called as intermembrane space. Inside the chloroplast, there are flattened vesicle-like structures are present. Those flattened vesicle-like structures only we call it as the thylakoid. We got the point clearly. And we discussed a group of thylakoids called as granum. One group of thylakoids can be connected to the another group of the thylakoids by a thin thread-like structure, which we generally call it as the lamella. No point of getting confused here. And very important statement is what? It also contains a fluid-filled structure, which is called stroma. It contains the ribosomes, which are involved in the synthesis of protein. It contains DNA. I hope every student remember what is the major protein that can be synthesized by the chloroplast DNA. I believe everyone remember the major protein synthesized by this chloroplast DNA is said to be the rubisco, which is the most abundant protein on this planet Earth. The most abundant protein on this planet Earth is the rubisco, which is synthesized by the chloroplast DNA. Now, friends, what else the chloroplast contain? Chloroplast also contain various lipid droplets. Chloroplast also contain starch granules. But what is the point that I want you to remember? If you observe the starch granules, friends, starch granules are very smaller. Very, very, very smaller they are. But if you look into this particular starch granules are very larger, whereas lipid droplets are very small. But what you can observe is compared to the starch granules, you can observe more lipid droplets. Lipid droplets are basically storing the lipid, whereas friends, starch granules are especially involved in storing this particular starch. This is the simplified structure of the chloroplast. But friends, it is very important to understand deeper anatomical structure also. Why we have to understand the deeper anatomical structure? What is the reason? The reason is that inside the chloroplast, remember my words, inside the chloroplast, various reactions inside the chloroplast, various reactions takes place at various regions. At various places or various regions, various reactions will be taking place. That's the reason it is very important to remember every single region. Now, what do we call it as? A group of thylakoids we generally call it as friends granum. Now you all people look into this. This is one single thylakoid, is it? Now every one of you remember my words. This outer membrane of the thylakoid, what we generally call it as the the outer membrane of the thylakoid, we generally call it as a thylakoid membrane. Widely, we call it as lamellae. Widely, you call it as what? Lamellae. Don't get confused. And remember my words all the time. Any organelle, if it contains certain internal space, remember my words very correctly. Any organelle, any kind of a organelle, if it contains certain internal space, that internal space, we generally call it as the lumen. Whether it is a chloroplast, whether it is a mitochondria, doesn't matter. Any organelle, if it contains the inner space, that is what generally we call it as the lumen. Now remember, the entire lesson of the photosynthesis, the entire lesson of photosynthesis is dependent on this specific slide. That's all. If you understand this slide, most of the concept is very clear. Photosynthesis topic seems to be very easy, but trust me, 90% of the students cannot answer the question because of a tricky statement that they raise in the exam. So you require a clear-cut concept. Now, what is photosynthesis? Photosynthesis is a process of reduction of the carbon into glucose. That is clear point. We have to reduce the carbon into glucose. That is very clear-cut point. But friends, if any plant, remember my words, if any plant want to reduce the carbon into glucose, it is not that much easy process. First, plant require a lot of energy. Whenever they are having abundant amount of energy, they can easily reduce the carbon into the glucose. Now, the question is very clear, from where they get energy. So if they want to produce the energy, they will use the light. They will use the light. By using light, they produce energy first. Once energy got produced, then fixing the carbon into glucose is an extremely easy process. No point of getting confused over there. Now, why did we use the word light reactions? We used the word light reactions because they always occur only in the presence of light. If no light, it never occurs. That's the reason we call this particular light reactions as the light dependent reaction. They are extremely dependent on the light. No point of getting confused. Then what is their ultimate aim? What is the meaning of production of the energy? Which means that there is a production of ATP as well as, friends, there is a production of the NADPH also. No point of getting confused over there. So remember, what is ATP here? Energy precursor, major energy precursor. You should not use the word that NADPH is the energy precursor. No, you should not use that. You should always remember NADH is acting as a reducing equivalent. Very important point. Even you can also remember a word that it is involved in the transfer of the electrons. No point of getting confused over there. And friends, what are dark reactions? Again, I'm telling to all of you friends, dark reactions doesn't mean that they are occurring in the dark. Dark reactions means it doesn't matter whether the light is present. It doesn't matter whether the light is absent. Dark reactions always occur. If you recall, we eat the food morning time also, we eat the food evening time also. Similarly, when it comes to the plant, please mute your mics. Plant can prepare the food during the daytime also. Plant can prepare the food during the night time also. So what is the point you have to remember? It doesn't matter whether the light is present or absent. Then what has to present compulsory? Energy has to present compulsory. When energy is present, by using that energy, plant is going to convert the carbon dioxide into the glucose. Since it is not at all associated with the light, the dark reactions are generally said to be what reactions, friends? We generally said it to be the independent reaction. There is no point of getting confused over there. Now let's understand. Again, remember my words. Light reaction it is always light dependent. Since it is light dependent, when it will occur, friends, it will occur only in the daytime, only in the daytime. Then when you look into the dark reactions, there is no requirement of the light, which means it occur in the daytime also, it occur in the night time also. Dark reactions doesn't mean that they are occurring in the dark. Dark reactions means they are light independent reaction. It doesn't matter whether the light is present or absent. No point of getting confused over there. Now listen, light reactions are also said to be what reactions? Light dependent reactions. No point of getting confused. Okay. Now listen my statements. Very important. I told you, right? Locations. What is the goal? Never forget the goal till the end. Our goal is production of ATP, production of the NADPH. This is very, very clear. Now, where this light reactions are happening? The light reactions are happening. See, this is what we can call as thylakoid. Correct na? So on the plasma membrane of the thylakoid, the light reactions will be taking place. This is the membrane. This is what you can see is the membrane. On this membrane, the light reactions are taking place. Now the question is very clear. Why? Why light reactions are only happening on the thylakoid membrane or the lamellae? Why not in any other place? The reason is that all photosystems present here. Correct na? All electron acceptors present here. The most important, the presence of the ATP synthase enzyme. Since all are embedded within the thylakoid membrane, that is the reason we can say that the site of the light reactions are always thylakoid membrane or the thylakoid lamellae. Okay. After this, in the tomorrow class, I will show you the set question. The set question is also almost equal to the net question. So much confusing question. I believe you people will answer that question as well. Okay, listen. Now, what is our ultimate goal, friends? Our ultimate goal of the light reactions is production of ATP and NADPH. So in simple words, we have to produce the energy. So friends, who is involved in the production of energy? Undoubtedly, friends, photosynthetic pigments are involved in the production of energy. You people has to remember compulsory. Every one of you remember the concept. What is the ultimate goal of the photosynthetic pigments? They are the one which are generally converting light energy into chemical energy. That is the ultimate goal. Light energy into chemical energy only it is doing, nothing else. Now, all the photosynthetic pigments that are present in the plant and lower organism broadly categorized into two major types. They'll ask a very simple question, but if you don't know the concept, you'll answer it wrong. So all the photosynthetic pigments broadly categorized into principal pigments. Then friends, accessory pigments. So remember everyone, the principal pigments, friends, we also call it as antenna pigments. No point of getting confused over there. I'm so sorry. Principal pigments are also called as core pigments. Accessory pigments, we also call it as the antenna pigments. That is true. Okay. Now, listen, every one of you. All these principal pigments are broadly categorized into three types. So no point of confusing. Number one is called chlorophyll A. Number two, call it as bacteriochlorophyll A. Number three is called as chlorobium chlorophyll. So friends, what is the major fundamental difference between this chlorophyll A, bacteriochlorophyll A, as well as the chlorobium chlorophyll? Now every student understand. Chlorophyll A, remember everyone, it is the most abundant type of the chlorophyll in the green plant. That is also asked statement, exclusively present in the higher green plants. And very important, friends, bacteriochlorophyll A. Now, the question is here, what is the difference between the chlorophyll A and bacteriochlorophyll A? Remember my words. Whatever the chlorophyll A that gets modified, which is present in the purple bacteria, that is only we call it as the bacterium chlorophyll. In simple words, listen my statements everyone. Modified chlorophyll A molecule that is present in the purple bacteria, That is what exactly, friends, we can call it as the bacteriochlorophyll A. Similarly, the chlorophyll A, after modification, if it is present in the green sulfur bacteria, simple words, modified chlorophyll A present in the green sulfur bacteria, friends, that is what exactly we can call it as a chlorobium chlorophyll. No point of getting confused over there, and it is very, very easy to understand also. Now all the students listen. Accessory are antenna pigments. Very important, friends. What are those? Majorly chlorophyll BCD, carotenoid, and a very, very important one is the phycobilins. What I told you just now, we discussed that, friends, chlorophyll A most abundantly found only in the green plants, whereas chlorophyll B, C, D, they are present in the lower organisms such as the green algae. Now remember, friends, this carotenoids are basically of two types. Number one, we call it as the carotenes, and number two, we call it as the xanthophyll. Now, friends, what is the significant difference between the carotenes and xanthophylls with respect to the structure? Every student remember, carotenes basically contain only carbon and hydrogen, nothing else it contains. When it comes to the xanthophyll, along with the carbon and hydrogen, xanthophyll also contain the oxygen. Don't get confused. So that is the simple difference between the carotene and xanthophyll. What is the best examples of the carotenes? You can take a multiple example. You can take beta carotene as a very good example, which is present in the carrot. And very, very important, you can take lycopene, which is present in the tomato. They are basically what belongs to the category of carotene. The best example of the xanthophyll that you people can take it is lutein. I want you to compulsory remember for the exam. Okay, now the third category of the accessory pigments, friends, we are generally calling it as the very good zeaxanthin, phycobilins. So, friends, these phycobilins are most abundantly found in the cyanobacteria as well as the rhodophyta also. Now, remember everyone, what are the best example? We can say phycoerythrin, phycocyanin, as well as, friends, L-phycocyanin. I want you to remember every single example for the exam point of view, because remembering the examples only, major core concept here. Now again, the simple asked statement is, among the chlorophyll, carotenoid, and phycobilin, which pigments are a lipid-soluble pigments, which pigments are a water-soluble pigments? That is a very important statement to be remembered for the exam point of view. Always remember, friends, all the type of a chlorophyll molecule, all the type of a carotenoid molecule, both are extremely lipid soluble in nature. When it comes to the phycobilin, this is the only pigment, friends, which is extremely water soluble in nature. There is no point of getting confused over there. It is easy to understand. Now, friends, what is the structure of the chlorophyll? Is it really necessary to remember the structures, friends? As I told you, right, remembering the structures is actually important. Okay, you have to remember. Just give me one moment, please. All right, one second. I get the question here. 1 second, please. All right. All right, look into this. So it's very important to know the structure. If you observe the structure very clearly, now let's focus on the structure of chlorophyll molecule. I don't encourage even a single person also to remember the structure. I encourage every one of you to understand the structure correctly. If you look into the structure of the chlorophyll contain porphyrin head, very important point. Chlorophyll contain a phytol chain. Just now you remember my words. Just now I told you that the chlorophyll molecules are lipid soluble. What is the reason? You can see this. It is having a long hydrophobic chain. You can see that. It is having a long hydrophobic chain, which is a phytol in nature. Now you can ask me, sir, what exactly this particular tail will do? All the students remember, this is the thylakoid membrane, right? This tail will help this, but please mute your mic. Please, who is this man? Bhavani, mute your mic. Rajkumari, mute it. Okay, now listen everyone. So what I'm trying to explain to all of you, this particular tail of the chlorophyll will embed. This is a very important point. It will embed or it will incorporate the head into the membrane. Now the question is very clear. How a chlorophyll molecule is embedded in the thylakoid membrane? That is the question. So chlorophyll molecule embedded within the membrane with the help of tail region. There is no point of getting confused over there. Now that is called phytol chain, which is a long hydrophobic chain. And friends, what is this is called as head of the chlorophyll. Head of the chlorophyll, we call it as the porphyrin. What is porphyrin? How to understand this particular porphyrin actually? Listen, it contains four rings. You can see that A, B, C, D rings. Each ring is called as the pyrrole ring. Now tell me friends, how many rings are there total? There are total 4 rings are there. Every student remember 4 pyrroles. I want you to understand friends. 4 pyrroles come close together with the help of a nitrogen. They are surrounding central metal atom, which is the magnesium. I'll repeat the structure. No point of getting confused. Porphyrin ring means it contain a pyrroles. How many pyrroles? 4 pyrroles. We can call tetrapyrrole. So tetrapyrrole come close together with the help of a nitrogen linkage. It is surrounding the central metal atom magnesium. This is what exactly we called as porphyrin head. Now the most important point to be remember for the exam, as we are discussing from the starting, chlorophyll is the major pigment which absorb the light energy. Now the question is here, how chlorophyll can absorb the energy? Chlorophyll is absorbing the energy just because of the presence of the magnesium. Why friends? Here magnesium which is present in the chlorophyll, that is the one which is generally acting like a chromophore. It is acting like what? A chromophore. Don't get confused with the point. Now all the students try to understand. Just now we discussed, we have a chlorophyll A, we have a chlorophyll B, we have a chlorophyll C, we have a chlorophyll D. Now understand my statements very correctly. All these chlorophyll molecules having the same head, porphyrin, same tail, phytol chain. Now the question is very clear, how we are going to differentiate one chlorophyll molecule with the another chlorophyll molecule. That is the crucial point here. One chlorophyll molecule can be differ from the another chlorophyll molecule, friends, exclusively based on their side chain. You can consider solubility is also one of the parameter, but it is not a major parameter. But majorly we will consider really based on side chain. If you look into the chlorophyll A, it is having a methyl group as their side chain, and if you have a chlorophyll B, it is having aldehyde group as their side chain. Suppose if you look into the bacteriochlorophyll, it is having something else as their side chain. No point of getting confused over there. Now question is super clear. One chlorophyll molecule can be differ from another chlorophyll molecule exclusively in their side chain only. Now the next and very important simple structure, that is the carotenoid. And friends, how this particular carotenoids can be different from each other, that's a very important statement. All the students focus on my words. Carotenoids can be differ from chlorophylls, especially based on their structure, which is called as a isoprene structure. What is the meaning of a isoprene structure? Every student remember, isoprene means, remember my words, 5-carbon containing compound. What I'm saying, it is just a 5-carbon. 5-carbon containing compound with the alternate single bond, double bond, single bond, double bond, single bond, double bond. What friends? So 5-carbon containing compound with alternate single bond and double bond, we generally call it as the isoprene unit. Don't get confused with the point. Okay. Now, bilin pigment. Very good. Okay. Okay. Now tell me, Shafia, very correctly. Tell me, Selvi, carotenoids are actually contain how many carbons? Carotenoids are generally contain how many carbons? Very good. Very good. Very good, Swati. Carotenoids are basically what? Tetraterpenes. They generally having how many carbon? They are generally having the 40 carbons, which we generally call tetraterpenes, because the carotenoids are comes under the category of the terpenes. Don't get confused with the point. All right. Now, friends, what are bilin pigments? No point of getting confused. Bilin pigments, we generally call it as what, friends? We generally call it as the linear tetrapyrrole. Now, question is super clear. Here, chlorophyll A, what we said? Chlorophyll A, we said that it is a circular. But here, what we said, the linear tetrapyrrole. Linear tetrapyrrole, we generally call it as a linear tetrapyrrole means one pyrrole, second pyrrole, third pyrrole, fourth pyrrole, but they are linear, which is a straight chain. That's what we can call it as the linear tetrapyrrole. No point of getting confused over there, and it is easy to understand. It is clear. Now, you understand the structure, but what is the very unique statement? The unique statement here is that we all people know that chlorophyll pigment absorb the light energy, carotenoid absorb the light energy, bilin pigments are also absorb the light energy. Now the question is that if everyone is absorbing the light energy, what is the use of the three pigments in the plant body? So one of the reason is that even three are absorbing the light energy, they can absorb the light energy at a different wavelengths. At different wavelengths. No point of getting confused. Not only that, very important point, compared to the chlorophyll, compared to the bilin pigments, carotenoids are the one which are protecting the plant from the excessive sun, or in simple words, we can say that carotenoid is the one which are protecting the plant from the photooxidative damage. No point of getting confused here. Very, very good. So what is the role of the carotenoid in the plant? Very good. Carotenoids are actually acting like a scavengers, means which are preventing the excess of light that can be absorbed by the plant. Don't get confused with the point. Now, it is super important. What is the common point? The common point of the three pigments is they are absorbing the light. Now, all the students try to understand me. This is the question which we got in the 2023 paper. Based on the absorption of light, every student try to understand me. Based on the absorption of light, there were two spectra were derived. One is called as the absorption spectra and another one is called as the action spectra. And friends, what is the clear-cut difference? What is the clear-cut difference between the absorption spectra and between the action spectra? I'll tell you in a simple words. No point of getting confused. Absorption spectra means, understand my words, absorption spectra always explaining about pigment. Always it is explaining about pigment. What it is explaining about the pigment, at which particular wavelength, what I'm saying, at which particular wavelength, the particular pigment absorbing maximum amount of the light, that is called as absorption spectra. What, friends? At which particular wavelength the particular pigment is absorbing the maximum amount of the light, that is what exactly we can call absorption spectra. Now, if you ask me a question, does every pigment having the same absorption spectra? That statement itself wrong statement. Every pigment do not have the same absorption spectra. All the students, please mute your mics. Please mute your mic. Anand, mute your mic. All right, look into that. Look into that. So what I'm trying to explain you here, different pigments are going to absorb the light, at a different wavelengths. On the x-axis, we have taken wavelength of the light. On the y-axis, we have taken the amount of the light absorbed. Every one of you focus on the picture so clearly. Okay. First, we have taken a different pigments. We have taken a chlorophyll A, we have taken a chlorophyll B, and we also has taken a carotenoid. Look into the picture. Look into the picture. Now, if I observe the chlorophyll A, look into this. Chlorophyll A, see, the peak is very high till this range. What I mean to say, 400 to 500 nanometer, the chlorophyll A absorbing the light at very high speed with a good wavelength also. After that, the peak was fall down. So what I have to explain here? I have to explain here, chlorophyll A absorb dark blue light. It absorb light blue light also. But friends, after that, it is completely, coming down. So what does it mean? Remember my statements, all the students. Whenever a color absorbed by the pigment, if a pigment absorb a specific color, it never appear in that color. What, friends? Whenever a pigment absorb a specific color, it never appear in that color. But friends, whenever a pigment reflects a color or rejects a color, it always appear in that color. So you can observe the picture very clearly here. What you can see here, chlorophyll A, what it is doing, it is absorbing the dark blue light, so it never appear in the dark blue color. It is absorbing light blue color also, so it never appear in the light blue also. So, but chlorophyll A, very greatly, it is reflecting the green light at a higher fractions. So all the plants appears to be green in color. The reason is that the chlorophyll A always reflects the green light. No point of getting confused over there, and it is very, very easy to understand also. All right, that is clear. Now all the students try to understand my statements correctly. If you look into this particular carotenoid, friends, especially carotenoid absorbing dark blue light, carotenoid absorbing light blue light, carotenoid absorbing a green light also, but it is not absorbing yellow light, it is not absorbing orange light, it is not absorbing red light. Now, if you see the best example of a carotenoid, we can take an example of a lycopene. Lycopene is what actually it is present in the tomato. So why the tomato is red in color? Because the lycopene carotenoid is reflecting the red light at higher fractions. It won't absorb even a little fraction of the red light also. It completely reflects it. That's why tomato appears to be red in color. Now, if you look into the carrot basically contain what? Carotene. So carotene is always reflecting the orange color. That's why all the carrots appears to be orange in color. You might observe some of the carrots red in color. If you go to the Ooty, Kodaikanal, they will freshly pluck all those particular carrots, along with the roots, and they will sell. You might observe it. Correct, na? Actually, those type of carotene, they can they can reflect slightly red color. Slightly, they can reflect the red color. Right. So, but most of the carrots are orange in color. Some carrots even they're red in color. So everything is just basically because of what? Because of the type of the pigment that it contains. So in final words, whatever the color absorbed by the pigment, it never appear in that color. But whatever the color reflected by the pigment, it always appears in that color only. Never get confused with the point. That is what we can call absorption spectra. Then what do we can call action spectra? See, very simple to understand. Absorption spectra, it is always talking about what? Pigment. It is always talking about the pigment, at which wavelength the pigment is absorbing the maximum light. This is what it will talk. Now, if you look into the action spectra, it is always talking about a process. It is always talking about a process. So any process, friends, doesn't matter. Any process, at what wavelength the process is happening at its maximum. Tell me, Selvi, very clearly. Akila, tell me, Kalyani, Anand, at what wavelength the photosynthesis occurs at a maximum rate? In other words, what is the most suitable wavelength for the process of the photosynthesis to occur? So why only 600 to the 700 nanometers? Because at a 600 to 700 nanometers, friends, most of the pigments absorb the light. Most of the pigments absorb the light, okay, which is actually required. Okay, so actually we can say that 400 to 800 nanometers, but most suitable wavelength for the photosynthesis is 600 to 700. Why? Because PS1 and PS2, they are actually absorbing the light at these wavelengths only. That is one of the major reason. No point of getting confused over there, right? Which is 680 and 720. So what is the conclusion? Absorption spectra is actually talking about at which particular wavelength the specific pigment absorbs the light, whereas action spectra is explaining at which particular wavelength the specific process occurs maximum. Here an example of the process I have taken, photosynthesis itself. So it is getting occurring maximum at the rate at 600 to 700 nanometers. No point of getting confused over there. Now we will discuss. Please don't confuse with our goal. Our goal is production of ATP and production of NADP. Still, we have not discussed anything about our goal. Now, still we are just discussing about the structures only. All right. Now look into the picture very correctly. Yes, undoubtedly, Swati. Different depending on the pigments, undoubtedly wavelength. No, we can't say that the wavelength is getting changed. You can say that because different pigments are going to absorb the light at a different wavelengths. They can reflect at different wavelengths. We can say that. There's no doubt about it. All right. Now, let's look into it. Very important, friends. Every student focus on this picture because this is the most important picture everyone has to understand. If you understand this, almost the light reactions will be little clear. This is what, friends? Thylakoid membrane. On the thylakoid membrane, what is present? On the thylakoid membrane, photosystem is present. Inside the photosystem, what are present? It contains LHCs, light harvesting complexes. Inside the LHCs, what are present? That is important, friends. Inside the LHCs, we have reaction center, we have antenna pigments. You should always remember this point. On the thylakoid membrane, first photosystem is present and photosystem contain LHC. Inside the LHC, reaction center and antenna pigments are present. And every student commonly know that reaction center always contain chlorophyll, nothing else it contain. Reaction center always contain the chlorophyll. And remember, antenna pigments always contain chlorophyll B mostly, then carotenoid, then xanthophylls. Don't get confused with the point. Now understand. So this is the thylakoid membrane. This complete thing is called as the photosystem. And friends, here you can see this, right? These are said to be the LHCs. These are said to be the LHCs, which contain antenna pigment. And friends, this is what exactly we can call reaction center. Don't confuse. You'll understand in the upcoming slide. Now listen, I'm going little slowly. You have to understand. But tomorrow I don't go this much slow. Okay, because it is a known topic for everyone. Why we always say P680 or photosystem 2, and why always we say P700 or the PS1. The reason is that photosystem 1 is the first discovered. That's why we can call PS1. Photosystem 2 is the second discovered. We can call that as the photosystem 2. But what we discussed, any kind of a photosystem, reaction center basically contains what? Chlorophyll A. So friends, in case of a photosystem 2, the chlorophyll A, which is present in the reaction center, it is absorbing the light at 600 nanometers at a maximum intensity. So we can call P680. It is the 680 actually coming from chlorophyll A, because the chlorophyll A is absorbing only at this wavelength at higher fractions or at higher intensity. Whereas photosystem 1, here also it contains the chlorophyll A, but the chlorophyll A basically absorbing the light at 700 nanometer. No point of getting confused over there. It is easy to understand here. All right. Just try to listen to me very correctly. If you got this, if you understand this concept, it is very clear. This concept even present in the NCERT and academy books and everywhere. But most of the people, don't understand this diagram very correctly. So this is the diagram generally explaining us how exactly energy can be produced. Understand. These are all what pigments? These are all said to be the antenna pigments, made up of chlorophyll B, made up of xanthophylls, made up of carotenoids. Now, what's the function of them? The function of them is absorption of the light. Listen, look into every one of you this. This dark green color one, that is what we generally say that reaction center. What I told to all of you, reaction center mostly made up of the chlorophyll A only. So look into this, look into this. Now, all the antenna pigments absorb the light. Now it is passing from one antenna pigment to the another antenna pigment. Finally, they are passing the energy to where? Finally, they are passing the energy to the reaction center. So friends, passing of this energy from one antenna pigment to the another antenna pigment, we generally call it as resonance transfer of energy. Never ever get confused with the point. What do we call it as? Resonance transfer of energy. Now, you should always remember with respect to the chemistry, when it comes to the electron, always remember my words, always remember this. In biology also you remember like this only, especially biochemistry. When we lose the electron, we call it as oxidation, oil, rig. When we gain the electron, we call it as the reduction. Now you only answer my question. First, what is going to happen? This antenna pigment gain the electron. When it gain the electron, reduction will happen. Now immediately it won't keep the electron with it. It is going to pass the electron to the next pigment. Then what do we call it as? We call it as it got oxidized. It got oxidized, but someone received it, right? Then it will become reduced. So what my point here? My point here is that on the antenna pigments, several redox reactions takes place. What are redox reactions? A coupled oxidation also, a coupled reduction also. So that oxidation and reduction together, we generally call it as the redox reactions that are exclusively happening on the antenna pigments. Look into that, look into that. See, antenna pigment absorb the photons and transfer it to the reaction center. Now friends, finally energy came to where? Finally energy came to the reaction center. Now you tell me, suppose if you had instant glucose, tell me what happens to you. Suppose example, you had a coffee just now you had a filter coffee. What happens? Your CNS will be super stimulated. You will be very much excited, at least for the period of 1 hour. Why? Because CNS is getting stimulated because the coffee contains a caffeine. Similarly, friends, what happened here? When the energy transferred to this reaction center also get excited because it received a lot of energy. Whenever it got excited, friends, it is going to release excited electrons. Such an important and unique point it is. What it is releasing? It is going to release the excited electron. Now remember my words, that excited electron is accepted by a several electron acceptors. No point of getting confused. That particular electron is accepted by several electron acceptor. Don't think that only one electron acceptor is present. It starts with one. Sometimes it contain 10, even sometimes it contain 5, 6, 7. It depending on the type of the photosystem. Now, this is the most important. Let's consider this is the first electron acceptor, second, third, fourth, fifth, sixth, and so on. So friends, understand. Whenever the electron is moving from one electron acceptor to the another electron acceptor, what happens? Every student remember my words. Whenever proton moves, energy will be generated all the time. All the time you remember. Whenever electron move, energy will be lost actually. And someone will gain that energy. So similarly, when the electron is moving from one electron acceptor to the another electron acceptor, it lost certain energy. Friends, that energy we used to combine ADP with PI, hence there is a formation of the ATP molecule. This is how we produce the ATP, because the ultimate goal is what? To produce the ATP. So every one of you understand it. We have to continue it also. Antenna pigments absorb the photons and transfer it to the reaction centers. This is what we can call the resonance transfer of energy. Then what is going to happen? Reaction center get excited and release high energy electron. Now, who is going to accept that electron? That electron is transferred through a series of electron acceptors, and whenever it is moving from one electron acceptor to another electron acceptor, there is a loss of the energy. That energy can be used to produce ATP. Now always remember, electron always travel downhill. So where one is gaining the electron, one is losing the electron. That's why we can say that oxidation reduction reactions. Don't get confused with that. Now let's understand the same concept more elaboratively. What is our goal? Our goal is to produce the ATP, to produce the NADPH. Okay, I don't want any one of you to make the mistakes. Okay, listen. The production of ATP generally we can call it photophosphorylation. Question is very clear, friends. Why do we use the word call it as photophosphorylation? Phosphorylation is a process where ADP molecule combined with the PI molecule. By that, there is a production of the ATP. Now, the question comes here, how does the ADP combine with the PI? Is it that much easy? No. If the ADP want to combine with the PI, friends, there is a compulsory requirement of the energy. Now, from where that energy is coming from? That energy is coming from the sun. That's the reason we use the word photophosphorylation. No point of getting confused over there. Okay, photophosphorylation is the conversion of the ADP to ATP using the energy of sunlight. So now this particular photophosphorylation, depending on how electron is getting transported, we categorize them into two types. One is called cyclic phosphorylation, another one called, friends, non-cyclic phosphorylation. Very important, unique statement has to remember. Tell me, Anand, very correctly. Among the cyclic and non-cyclic, which kind of a phosphorylation is more predominant in the bacteria? Everyone of you answer. Which is more predominant in the bacteria? I'm talking only specifically bacteria. So specifically in the bacteria, cyclic photophosphorylation is the most predominant. Whereas non-cyclic photophosphorylation, friends, it is exclusively taking place in the higher plants. No point of getting confused over there. Okay, it's very easy to understand. And in the cyclic photophosphorylation, always remember there is only one photosystem is involved. In case of a non-cyclic photophosphorylation, friends, always there is an involvement of the two photosystems. No point of getting confused over there. One photosystem here, two photosystem here. Predominant in bacteria, predominant in the higher plants. Now, all the students pay attention. No point of getting confused. First question, why did we use the word cyclic photophosphorylation? What is the reason why I have used that word? Because the movement of electrons are in cyclic manner, which means that wherever the electron started moving, it is reaching to the same point. That's the reason we can call it cyclic photophosphorylation. All the students look into the picture. Antenna pigments are absorbing the light. They are sending to the reaction center. Reaction center get excited and it release the excited electron. Who is going to accept that excited electron? That excited electron is accepted by ferredoxin. It is accepted by plastoquinone. It is accepted by cytochrome, B6F complex. Finally, it is accepted by plastocyanin. But what is the point here? Am I audible to everyone? I hope I am audible. Okay. Now, from the plastocyanin, the electron is once again moving directly to this particular origin only. So in a simple words, friends, wherever we have started, the electron reached only to that area. That is the reason only we call it as the cyclic photophosphorylation. So it is very important. You all people has to remember the series of electrons. Number one is ferredoxin. Number two is plastoquinone. Always remember, friends, plastoquinone differentiated into plastoquinone A as well as the plastoquinone B. And we are having a cytochrome B6F complex. Then we are having the plastocyanin. From the plastocyanin, it is coming here only. Now you understand. Whenever the electron is moving from one electron acceptor to the another electron acceptor, there is a loss of energy. That energy can be used to join the ADP with the PI. Hence, there is a formation of ATP. Now, there is a very simple question. Answer, everyone. Do we really fulfilled the goal of light reactions by using cyclic photophosphorylation or not? Do we really fulfilled the goal of the light reactions? No, because the goal of light reaction is production of both ATP also, production of NADPH also. But we produced only ATP, we did not produce NADPH. So because of the absence of the photosystem. Okay. I hope it's clear for everyone till this particular slide. Okay. I hope it's clear for everyone, and requesting every student, read a little faster. Okay. Because these topics are easy, and tomorrow I don't teach this much slowly. I'm telling you, I'll go very super fast. Okay. That's the reason I want you to revise on the same day without confusion, because I'll go little speed from tomorrow class. Okay. All right. Okay. Thank you, everyone. Every one of you join at a sharp 2:30 p.m. I'll share you the link. Once the 2:30 p.m. class over, I will give you all the four recorded classes. Okay. And Swati Pheophyton is present in the non-cyclic. That is present in the non-cyclic. We will see that. All right. Everyone join at sharp 2:30. Thank you all.